All right, so we are talking today about phylogenetic systematics, and you have two labs coming up on this subject. So this will be an introduction to the terminology, the history of how we arrived at this revolution in systematics in the late 20th century and where we stand today, and then the basics of phylogenetic systematics, and this should feed back with your lab. Your lab will go into greater detail on the subject than we will in here. I need to finish with our discussion of speciation from last time, and then I will carry on with phylogenetic systematics through Wednesday, and by the end of Wednesday we'll be caught up on all of that. We were discussing speciation, which was the main phenomenon of interest to Darwin and many of his contemporaries. That remained a source of study, of course, right through today, and we talked about the modern synthesis as a unification of Darwinian ideas and insights, new insights in genetics, so in population biology. One of the long-standing discussions around speciation concerned the geography of speciation, whether speciation happens within a single place or whether it happens when populations become divided. According to the biological species concept, reproductive isolation is the heart of speciation. Barriers to gene flow between two populations create the conditions via which species can form with integrity and maintain their integrity separately. So geographically, how does that occur? From an original population of orange-shaded fishes, you may imagine that the pond or lake in which they reside is split into two, creating a barrier to gene flow, creating, at least temporarily, reproductive isolation. Now, this may be a cyclical occurrence, and the waters will rise again and the lake will reunify. But if it doesn't, you have the conditions just geographically set to have the two populations diverge genetically through the now familiar forces of microevolutionary change. But if the waters don't separate completely, can the conditions exist here for one population to divide into two in the same place, sympatrically? And that's a running debate, allopatric versus sympatric mechanisms of speciation. And so just need to review some of those ideas here. Certainly there is plenty of evidence for allopatric modes of speciation. That's not in question. It's really a question of how prominent sympatric modes of speciation have been in the history of life. Great examples of allopatric speciation abound. And you can look, for example, to the desert Southwest and these little squirrels that are divided by a deep canyon and have differentiated on either side into two good species because of this barrier, the deep canyon and the waterway in between that has separated them. You'd call this a vicariance event because the inference is that there was once an original population that was split geologically by the formation of the ravine. And because of the geological split that formed in the middle of the population leading to speciation, we call it a vicariance event. A good example of allopatric speciation. There's sort of a continuum of allopatric versus sympatric speciation. You can imagine a population that's on the periphery of the range, not entirely separated, maybe just a small barrier, maybe a bit of overlap. This subpopulation maybe just by the thinnest thread divided geographically from the original population. And if you want a term, something in between sympatry and allopatry, peripatry, like periphery. A peripheral isolate. An isolated subpopulation on the periphery of the range can be an effective mechanism for a branching event of one species off of an ancestral species. Because if this subpopulation forms out here, think of what begins to happen. If it's a small subpopulation, you have genetic drift that's going to be happening there. You may have adaptation to local circumstances if the physical habitat or the other organisms with which it coexists differ here. You're going to have adaptation that changes the allele, the alleles in that population, changes the gene pool. Now, in a peripheral isolate, as opposed to a population that's deeply separated geographically, there still may be gene flow, as illustrated here between flowers on one side of this stream and flowers on the other side of the stream. You may have gametes that continue to flow between populations, and that can hinder the branching event. That can keep the species together as a result of the sharing of genetic material. So, especially according to the biological concept, you need to have a firm divide here to really advance the speciation process. Here's a nice model that I can introduce now, and you'll probably need to wrestle with it another couple times on your PDF to really get what's happening. It's a bit complicated, but I think it encapsulates a lot of this, what I've been saying here. So imagine an original population and its gene pool here, with gene flow readily occurring among the members. Through time, looking in this direction, here are a couple things that happen to this population. First of all, a geographic barrier forms, or some kind of barrier to gene flow. And that's the red line indicating that. So something gets in between the members of this population, reducing the flow of genes between some members of the population and other members of the population. Over time, these subpopulations begin to diverge through all the familiar mechanisms, to the point where they've diverged quite a lot, as illustrated by the change in color of the genetic components of these two groups. Now, gene flow might still be possible. They're sufficiently related that they recognize one another as mates, or the gametes remain compatible as a result of a continued shared similarity. And a couple of things might happen here. They may continue just in this way. They may just maintain this distinctiveness, diagnosably distinct at some level, genetic or morphological, but capable of forming offspring, successful offspring in a hybrid zone. And so essentially you've got three diagnosably different entities here. One here, the hybrids, and the original ancestral form. And that can be maintained more or less indefinitely. You see it in things, you see it in nature. Baboons, for example, in Africa, often show this pattern of two distinct types, and in the neighboring area where their populations join, hybrids that form, and as long as people have been watching, you have the two types and a hybrid form separately. So that can just be maintained. Or the two diverging populations could come back together and just reform that an original type, resemble the original type or come to look like something else through time, but they could fuse and come back as one entity. That's a possibility. You could imagine the barriers to gene flow breaking down and the two populations merging again. Or that those barriers to gene flow could get reinforced and the two groups could continue to diverge and speciate, forming two distinct lineages that stop crossing back and forth completely in the final steps of a speciation event. Or what I was describing originally, the stable hybrid zone maintaining its distinctiveness. So those are all possibilities in the process of speciation. And this is sort of where we've arrived at by the late 20th century in our understanding of Darwinian mechanisms, population genetics, and ongoing field studies. I say the late 20th century because that's That's where your textbooks stand, mostly. The really contemporary research continues to investigate this and continues to support and challenge all of this. Sympatrically, can organisms speciate? A lot of ink was spilled on this subject over the decades, and some of it unnecessarily, much of it interesting. But let's look at a couple of clear examples where certainly sympatric speciation seems to be in evidence. This humble little creature exists in a larval form as maggots that eat fruit of trees, and this species particularly of Rhagoletis fed in the natural conditions on hawthorn trees in the eastern United States. So the larvae would eat the fruit of these plants. They would burrow into the fruit of the hawthorn tree, and the adult flies would also feed on the fruits of the hawthorn tree. And that was a tree that grew throughout the northeast United States, and the fly was associated with that tree. Apples were introduced to these same areas in New England in the 1800s in widespread apple orchards. And what this fly did was jump from the previous host species, the hawthorns, to the apples, and came to live off of the apples exclusively. Much more recent studies from the last 5 or 10 years have shown that the flies that jump to the apple trees now are attracted to the odors of the apple fruits exclusively. They're not even attracted any longer to the hawthorn fruit odors. And the flies that continue to stay on hawthorn trees aren't attracted to the odor of the apple fruits. So you've had a divergence occur within the fly population as the host plant was switched to by some flies in the form of these apple predators and their problems for the apple orchards, of course, and the apple farmers. So that's happened all in the same place. That's happened sympatrically there in New England, in the northeastern United States. And this is where the ink starts to get spilled in these old debates. And I find it a little tedious, I must be honest, whether this is allopatric speciation or sympatric speciation. You have pretty clear evidence of a strong divergence in the population. But is it truly sympatric? Well, what's your scale of analysis? You always need to be sensitive to the scale, the spatial scale of your analysis, something that will be reinforced in the ecology section. Ecologists are really good at being sensitive to scale. The scale here is New England, a big area crossing a few states of the northeastern United States. On that scale, of course, this is sympatric. The hawthorn trees, the apple orchards, they're all in that area. But how about when you get down to a micro scale? For these little flies, even the scale of an orchard relative to a forest might signal a difference in geography. The orchard is a geographically separate space from the hawthorn tree forest, right? Or even at the scale of individual trees. A hawthorn tree and an apple tree just over here may be spaced widely enough to be considered allopatric from the perspective of the fly. So always consider the spatial scale and spatial extent and the perspective of the organism even when considering these notions of allopatry and sympatry. All right, let's try to get a more exact example of sympatric speciation. And you can find this in abundance in plants, the phenomenon of polyploidy, leading to speciation rapidly in plants. The fly example, that's rapid too. That only happened over the space of less than 100 years. That's really fast, according to Darwinian standards, to have an event lead to divergence that quickly. Well, this is an example of polyploidy. This can happen in a single generation. The mechanisms are not important to understand at the cellular level, the molecular level, but just understand that you have a chromosomal alteration within a generation's time, whereby the chromosomal complement of the organism changes from parent to offspring. It's duplicated or triplicated, such that a diploid organism, upon breeding, usually through some type of reproductive error in the genetics, produces offspring with more than the normal 2 sets of chromosomes. And it's happened a lot in plants. You probably heard about it in the last section, that polyploidy can account for much of the diversity in many groups of plants. The percentages are debated, but it's over half of plants probably that have speciated, higher plants that have speciated via mechanisms of polyploidy. And if that's just happening through genetic phenomena within an organism across a single generation, well, there's sympatric mode of speciation by any criterion, right? This little fern here, tiny little fern, there's a coin for scale. It can have over 1,000 chromosomes. All these chromosomes having built up through the phenomenon of polyploidy. Or a big radiation like raspberries, rubus, the raspberries, the big thorny bushes you see in the park that come into fruit later in the year, those are blackberries or raspberries, members of this big genus. And you can see here the ploidy levels of these different species on this in this legend here, showing all the different events that have led to the diversification of this group, just as a result of internal genetic changes within the lineage, having really nothing to do with geographic separation, but having a lot to do with reproductive isolation still, because chromosomal incompatibilities can create reproductive incompatibilities, and thus reproductive isolation. This is a cladogram, by the way, and we'll be examining what the heck this branching thing is in more detail next. And just a final example, in a conservation biology context, this can be important when you have 2 different species that can come together and share chromosomes across species in allopolyploidy, as opposed to autopolyploidy, which was what I was describing really up till now. Auto, self, allo, other. Allopolyploidy involves the combining of genes between 2 previously separate species. And that can lead to speciation. That can lead to the arisal of new forms, but it can also be a big problem in a conservation biology context. If you have one species come into an area previously occupied by a first species and have them combine genetic material to produce a new form, essentially rubbing out the original species and even the integrity of the invasive. And that's a problem for conservation biologists who work to maintain natural populations, natural, local, native populations of organisms. Allopolyploidy versus autopolyploidy. So nothing that you need to know in detail on the mechanisms there underlying all of that, just the existence of the phenomenon as a source of sympatric speciation. So getting into the phylogenetic systematics discussion and thinking about the overall tree of life, all the diversity of organisms on Earth, all those collections that were made through travels around the world, all those new specimens that came into the hands and laboratories of scientists, there was a need to organize all this stuff. You've been studying the plants, right? And the fungi. Those fungi, they look like yes, those look like mushrooms. Plants, fungi. You were introduced to some of the prokaryotes and some of the protists in the last section as well. Those are parts of the overall tree of life. Well, how do all these things fit together? How do you bring organization to all this diversity? Because there's unity to this diversity according to evolutionary theory. There's a derivation from a common ancestor, and we need ways to classify all these organisms. And that was part of what Linnaeus accomplished with his Systema Naturae. Particularly the later editions of the Systema Naturae helped to found the basis for modern classificatory systems. These are the systems that Darwin inherited later. Classification, the process of defining and ranking species within hierarchical groups. So it was a great hierarchy of organization, these categorization schemes. A hierarchy because the creator's intentions and ideas were hierarchically organized, according to Linnaeus. And so the hierarchy worked because it fit the creator's intentions. Linnaeus based his assignments of organisms to different categories based on their essential characteristics. He was after the essence of the organisms and these necessary and sufficient conditions in the philosophical sense for identifying them. A couple of you have told me you're reading Aristotle in other classes now, so maybe you have bumped into his arguments based on necessary and sufficient conditions. Linnaeus was using Aristotelian philosophical principles for identifying organisms. More basically, he was analyzing for something like plants the floral parts, analyzing in detail the reproductive parts of plants in order to determine who they were and to assign them. And he gave them the names according to his binomial system, right? Linnaeus was doing this in the 1700s, but his names are still with us for many familiar creatures. Your humble toad of Europe, Bufo bufo, still called Bufo bufo, and Linnaeus named it. Toad so nice they named it twice. The brown bear, or the bears in general, Ursus, Linnaeus' name. He gave it to the bear in the 10th edition of the Systema Naturae, and it stuck. House mouse, mus, passion fruit, mango, sea lettuce. You definitely saw sea lettuce in lab, right? Ulva. That's Linnaeus' name. It's fantastic. All those things are still with us, with an incredible amount of change as well. And of course, our own species, Homo sapiens, the wise people. That's us, as wise as we are. Linnaeus introduced those names. Just to remind you, we capitalize the genus and we use a lowercase for the specific epithet, and you put the name in italics or underline it. And just do it. Just learn it. Because you'll read the newspaper about a new scientific discovery, and the newspaper, the editors won't have caught the fact that the writer put the species name in capital letters, both the genus and the species. And maybe they didn't italicize it at all, and it looks totally amateurish. And you can get indignant and write an editorial and sort them out, please, because it's just the most basic step in scientific communication, hearkening back to Linnaeus. Now we're going to dismantle one major aspect of Linnaeus' classificatory system here in class. So we're not going to do his legacy any favors, but let's do his legacy one simple favor in this sense, in identifying species according to capital letter genus, lowercase species, italicize it. Maybe I'll put an exam question on just to make sure you're understanding that, because it's amazing how little understood that simple point is after hundreds of years of trying on the part of biologists. So brown bears, the local North American grizzly bear, Ursus arctos. The cave bear, Ursus spelaeus. Same genus, different species, right? Signaling that they're closely related in some sense. According to Linnaeus, because they share essential characteristics that are similar. Their teeth, I guess, or their skulls, whatever bones he would have had. According to today, the primary criteria are based in genes, although the morphology is still centrally important, and sometimes behavior and other features of the organism. Linnaeus' classificatory system was a hierarchy of ranks. And this is where we'll really challenge Linnaeus. His species, here, the leopard, Panthera pardus, this very accomplished cat of Africa and Asia. The leopard, a great hunter of forested systems, is Panthera pardus. And if you don't have room, you put a period after the first letter of the genus. So P period pardus. Or if you're just talking about the genus Pardus in general in a paragraph, just introduce Pardus at the beginning of the paragraph and use P period thereafter. You don't have to keep writing it. The context will let the reader know that you mean genus Pardus with your P period. Well, P. pardus, the leopard, is a member of the Panthera genus with a couple of other cats. They're all cats. They're all felids, members of the family Felidae. And members of the mammalian families have that suffix, I-D-A-E. So if you say something ide in the mammalogical context, someone will know you're dealing with a Linnaean family ranking. All carnivores or carnivorans, members of the order Carnivora. They're all mammals. They all have hair, and they all make milk for their young. They're all mammals according to those essential characteristics, and so forth, leading up to the kingdom of animals. And then an even higher ranking, which was introduced in the late 20th century to accommodate insights into the archaea and the differences of archaeans from bacteria, which we'll talk about briefly again in a moment. All of these are taxa. Each, no matter the ranking, it's a taxon. Chordata, that phylum, is a taxon. Felidae is a taxon. If you want to refer to all these taxa together, you change the suffix to the plural. If they're taxa, each is a taxon, no matter the level, no matter the rank. So much of that's familiar from high school biology, I think. And you had to memorize these ranks. Kings play chess on fine-grained sand, or something like that. Kingdom, phylum, class, order, genus, species, right? And you had to memorize that because that's so important, that order. What many biologists think now is that really it's not important, these ranks. This nesting hierarchy is certainly important. But what rank these levels exist on, whether it's a kingdom or a phylum, whether it's a family or a subfamily or a suborder, because there's lots of levels that have been named in between these major levels, those ranks, to some minds, including my opinion in general, are completely meaningless, and you can dispense with them completely. This hierarchical order is still important, but the rank, no. The names on the right, Animalia, Chordata, Mammalia, certainly important. Those identify your groups. Your more and more inclusive groups or your less inclusive groups, depending on which direction up or down the hierarchy you're going. But the rankings, no. Forget them if you want for our purposes in here. Your GSI might have a slightly different view, and certainly we welcome the different viewpoints. Something you should wrestle with, what we mean when we say, let's disband the ranking system of Linnaeus, but maintain the hierarchical organizational system for classification. Linnaeus' system worked in an evolutionary context, even though he wrote it in a non-evolutionary context. Darwin could reimagine the Linnaean hierarchy in a historical sense. There's your leopard here. Well, the leopard is just a branch of cat that diverged off of the other branches of the carnivora that includes things like badgers and otters and coyotes and wolves. And the coyotes and wolves are more closely related to each other because they shared a more recent common ancestor, according to Darwin, than either one is more closely related to any of these other organisms. Thus, you honor it with the same genus name, Canis canis, because it shares a more recent common ancestor. That's the evolutionary insight. That's the historicization of the classificatory scheme. And so it still works. Linnaeus' hierarchy still works in an evolutionary sense. You've just made it more properly historical. Systematics is this field of naming organisms and explaining evolutionary relationships. And it's a really important field that lacks fame and star power and financial resources. And so very few scientists go into that field for various reasons. And so there's a great shortage of systematists in the world. and there's so many organisms, such a backlog of organisms that need naming and description and their evolutionary history untangled, that it's a big problem in science. Not enough people describing fungi and flies and bacteria, doing that basic work in biology. And it's a big problem on a changing planet where so much is shifting and getting lost. Haeckel was a German who championed Darwin's ideas in Germany. And he introduced the term phylogeny and drew some of the first phylogenetic trees according to Darwinian ideas. He used an old symbolic like oak tree without its leaves as this metaphor for the branching history of life. He called this figure his pedigree of man. Man being on top of that figure. So this history of life that starts with the monera down here. What are monera? Anyone know that? The old word for bacteria. So the bacteria at the base, then there's amoeba here, and worms here, vermes. And then higher and higher you go. So you can also see the shades of a great chain of being in this first major tree of life. Low things at the bottom, the pinnacle at the top, man, all the way at the top of the tree, everything leading to that. So you see a lot of the ancient bias in a depiction like that, but it's an evolutionary depiction. Today, if you click on these information buttons, you can look at ways to explore the tree of life in a dynamic context on the computer. It's a really effective way on a computer to explore the tree of life, because a tree like this, which is reduced now from a big oak tree to these little branching filaments, same idea though, exists at different levels, scales, so that this big clade of flowering plants here can be seen at this level, but you can zoom in on one of these branches and have it open up into a bunch more branches at a finer scale. And this would correspond in the old system to Linnaeus ranks. This might be a class of organisms that you zoom in on, and then you get into the genus, and then you start to explore the species. And you do that effectively on the computer. So click on it and try if you want to explore the tree of life according to those ideas. This early study led to the recognition of three great domains of life, three major large groupings of life that exist at the base of the tree. And I won't belabor this. I think you received already some instruction on this. The two prokaryote domains and then the eukaryotes. And the whole phenomenon of endosymbiosis is part of this discussion. But I need to get into cladistics. Phylogenetic systematics, this revolution in systematics that began with the writings of an entomologist, an insect biologist, Willy Hennig, who insisted that we need to classify organisms strictly based on evolutionary history. And he rewrote our system for naming based on strict evolutionary principles. And it has become very influential. First, it was just important to museum scientists who were doing all of this hard work of systematics. Then it began to creep into the universities, and Berkeley had one of the leading roles in this. Berkeley started getting serious about this in the 1980s, 1990s, and so if you were a graduate student at Berkeley, you would have been introduced to cladistics much earlier than other graduate students around the world. And you'd go to a professional meeting back in those years, and you'd give a presentation, and you would start to talk about cladistics, and everyone in the audience would say, oh gosh, they must be from Berkeley. They're doing this cladistics thing, this weird cladistics thing. And really, it's caught on. So now I think the legacy, Berkeley can be proud of its legacy, and it's a champion of this new wave of phylogenetic systematics that has entered the textbooks. So finally, you get introduced to this now in your standard high school biology textbooks. But for many of you, this is probably brand new. And it's not easy. It's not intuitive. and you're going to have to wrestle with it in your labs for the next 2 weeks. Who has been introduced to cladistics through earlier biology classes? Good number of you. Look at that. Wow. That was well over half of you. According to these ideas, you define taxa based on common ancestry, and you diagnose taxa based on shared derived characters. Shared derived characters is a phrase we'll hear again and again. And a cladogram is a branching diagram of hypothetical relationships based on these ideas. So you can call it a tree, you can call it a cladogram, doesn't matter. When someone in the cladistic context refers to a tree, they just mean a branching diagram like this. This is just a really simplified version of that old oak tree of Haeckel's, though. Haeckel wanted a tree that branched into two things, branched into two others. A stick diagram accomplishes that without all of the bark and all of the other stuff. So it's really just a simplification of the same thing. But how you arrive at the building of that tree, how you construct that cladogram, is based on objective methods that help the field move along rapidly in a world where we have lots of species, lots of organisms to wrestle with. Okay, here's a cladogram and some basic definitions and terms we need in looking at the cladogram. We still have our taxa. So your branch tips out here represent taxa. And these could be at any level. These could be at, and I'll use the Linnaean terms for convenience, these could be at the scale of a genus. So these could all be genera. This could be Panthera and Canis and Taxidia for a badger and Lutra for an otter. Or these could be even phyla. These could be major groups of life. So you could do this for big groups or you could do it for small groups. They're all taxa. They're your branch tips. Two taxa that share a more recent common ancestor than either one does with another taxon in your depiction are sister taxa. So these B and C are sister because they share this recent common ancestor that's more recent to the two of them than any other organism has with them. These hypothetical common ancestors are nodes in the cladogram. This node here represents the hypothetical common ancestor of A, B, and C. If you trace the lineage C back and trace B back to their common ancestor, and then trace it back to the common ancestor with A, you arrive here at this node, which represents a hypothetical common ancestor of all three taxa. It's hypothetical because we may not even have any fossils to illustrate what this ancestor looked like. But we know it existed based on an evolutionary hypothesis. We may not know exactly what it looked like because there may not be fossils of it at all. There may be branches that occurred off of these ancestral forms that have gone extinct. So you can imagine this being a richer tree with forms that have gone extinct and no longer are in evidence. So this tree is a simplification of reality. It's a model, and it simplifies reality, and it's wrong. This tree is a hypothesis. It may be our best available hypothesis for the relationships of these among these taxa, but it's subject to revision. It's just every cladogram is a hypothesis, and just a hypothesis, subject to revision. But they're incredibly useful for doing work in biology. So here's the most recent common ancestor of all the taxa in question. These three taxa, well, we're not sure which one branched off first. Here, A branched off before B and C diverged. But here, we're not sure if D branched off before E and F or some other relationship. So they exist in a polytomy, and you get this forked structure because we don't have the sequence of relationships that led to those taxa. It's just a polytomy. The field of cladistics is very terminologically heavy. Willy Hennig loved complex polysyllabic words, and he made up a lot of words. He coined a lot of words to illustrate his ideas. So you'll have to learn some new terms in this process. Here's a tree with organisms, most of which are familiar: amphibians, mammals, lizards and snakes, crocs, birds. Bars over here illustrate the extent of the grouping. So the tetrapods exist starting here from this node, representing a common ancestor, and including everything from amphibians through the birds. These are all tetrapods. Tetrapoda, four-limbed. The ancestor of these creatures had four limbs. That little hash mark there, that little line, signals a shared derived character that's uniquely shared by this group. It's a synapomorphy. I'll put the word on the screen in a minute. It's one of these polysyllabic, annoying words. But synapomorphy, why don't I just write it now? S-Y-N-A-P-O, S-Y-N-A-P-O, M-O-R-P-H-Y. And you'll learn to love it, synapomorphy. It just refers to a shared derived character, like having four limbs. So everything that evolved from this ancestral form had four limbs, or lost those four limbs, like the snakes. Snakes don't have four limbs, at least that you can see. But based on this phylogeny, you can assume that their ancestor did, and something must have happened to those four limbs in the case of snake evolution. And it did. You find ancient snakes with limbs. Even the boas and pythons of today have little vestiges of the limbs in their bodies. Moving along the cladogram, you have another synapomorphy that diagnoses this clade, an amniotic egg with the basic structure of an amniotic egg anatomy, which amphibians don't have. And you can read that from the cladogram. If the amniotic egg evolved here, well, then the lineage is outside of that. The lineage is basal toward the base of the tree from that innovation. Don't have that character unless they evolved it independently. Amphibians don't have an amniotic egg. These reptiles do. Feathers. Feathers are a unique innovation, a synapomorphy of the birds. There are no other organisms on Earth with feathers that are not birds. It's a synapomorphy at that level. Sure, ostriches have feathers and hawks have feathers, but there are even other synapomorphies that distinguish the ostriches from the hawks and their allies. So you analyze these synapomorphies sequentially with greater and greater specificity to identify your lineages. Here is some of this terminology to use that illustrates some of the points I've been making. Let's think about that example of flying organisms and wings, right? Insect, mammal, bird, ancient flying reptile. It's not a dinosaur, actually. It's not a dinosaur, it's not a bird. It's a pteranodon. It's something different. Well, they all have wings, but they evolved them convergently. So they're not synapomorphies. They're not shared. They're all derived characters, these wings, but they're not shared among this group by virtue of having a common ancestor that had wings. No, the ancestor of this thing didn't have wings. The ancestor of birds didn't have flying wings. And the ancestor of the moths and the ancestor of bats. They were all flightless, the ancestors. So this is a convergent evolution, and characters that evolved convergently like that would be called homoplasies. Homoplasies, not synapomorphies, homoplasies. And you can get these shared similarities by convergence or reversion. I'll leave that for the moment. It'll be important in your lab. So looking at the detailed topology of a cladogram. When you think about the branching structure, you call it the topology of the tree. Look at A, B, C, and D here in this topological array of lineages. And think about their relationships. Which lineage is C more closely related to? As a result of a shared common ancestry, it's D, right? Well, how about this cladogram? Is it different in topology from the first cladogram? It's not. It's the same. Because look, who's the most common, closest relative of C? It's still D. And who's closest to C and D? Well, it's still B, just like it was here. You've just spun the branches around and shifted them a little bit. But the topology is exactly the same in essence to that first one. Same tree, drawn differently. You aesthetically have that choice. Well, I like this. It's prettier. I'm going to draw it this way. Okay, you draw it that way. That's fine. This one makes a lot more sense to me because it's so orderly and it's A, B, C, D. But it's your choice, your call, your aesthetic. But don't say that this tree is the same as these first two. It's not. Because look, C and B now share a more recent common ancestor than either one does with D. So this has a different topological configuration. It's a different hypothesis of evolutionary relationships. We need to name groups based on shared common ancestry, according to Hennig. The only groups that deserve names are those that include all the descendants of a common ancestor. The common ancestor being here, an appropriately named group would include all of these descendants. This box does not include a descendant in the form of G there. Thus, the members of that box don't form a good group, according to cladistics. It's a paraphyletic group and doesn't warrant a formal taxonomic name, according to the strictures of cladistics. A group that includes these taxa and lacks some of the common ancestors shared by these taxa, when you bring C into this grouping, is polyphyletic. You may be fooled into thinking this is a good group because C has convergent characteristics with these other members, or it has reverted to exhibit some of the primitive characteristics that diagnose these taxa. But a grouping like this is paraphyletic. Again, not a fair grouping, according to cladistics. The only good clades, the only clades worth a name in a phylogenetic systematics context are monophyletic groups that include all descendants of a common ancestor. A monophyletic group like that warrants a formal taxonomic name, and there are other clades, other good clades in this cladogram. D and E represent a monophyletic group. D, E, F, and G represent a monophyletic group. And then the whole lot of them, A, B, C, D, E, F, and G, are all a monophyletic group. Okay, I'll talk to you more about that on Wednesday. Thanks a lot.